focus of this video is on the role of regulated gene expression in controlling the steps that occur during development. So the basic question that we want to address in this video is how do we go from this, a fertilized egg, to this, a fully formed, complex, multicellular human being, right? That's the basic question that we want to answer. And during the process of development, we basically see two things happening. First of all, we see lots and lots of cell division, obviously. We start from one cell. We need massive amounts of mitotic division. This is something that we've talked about before. This cell cloning itself, essentially. This cell dividing by mitosis and all of its daughter cells dividing by mitosis to produce the billions upon billions of cells that make up this fully formed individual. The second thing that happens during development, which is more the focus of this video, is how those cells achieve specialized characteristics that are different from one another. So in other words, how do we get lung cells and neurons and liver cells and all the different cell types of the body? And this process is called differentiation. So all of the cells produced by mitosis, as we've talked about before, are genetic clones of one another, and yet we know that as a living organism, a multicellular organism, we're made up of different organ systems that are comprised of different organs and different tissues, those tissues being made up of multiple cell types, each with different characteristics that are specialized to carry out some function. So differentiation refers to the fact that we end up with cells that have different characteristics, and because they have different physical characteristics and properties, we can have different functions associated with those cells. And obviously the coordinated integration of all of those functions is what makes us as an organism possible. Just to make it perfectly clear what we mean by different characteristics, let's just take a look at a few cell types that we'd see in the human body and highlight some of the structural and functional differences that exist between these different cell types. So here are 4 types of cells that you'd see in every human being. Here we've got red blood cells, and if we look at the structure of red blood cells, what we can see is that they have this kind of flattened, round structure. And red blood cells are basically big bags of hemoglobin protein. Hemoglobin protein is a protein that's specialized to bind to oxygen, and that allows these red blood cells to carry out their function in the body, which is to transport oxygen through the bloodstream. Here we see neurons. Neurons are the cell type that make up the functional basis for the nervous system, and the nervous system functions in control and communication, right? So these neurons, in order to communicate, actually form these specialized structures where they send out these elaborate processes into their environment that make contact with neighboring cells, or sometimes even cells that are a large distance away. And those connections that are established between these cells allow them to communicate with one another. Here we see muscle cells, and as we know, muscle cells are specialized for contraction. If we zoomed in and looked at the intracellular environment within a muscle cell, we would see these arrays of cytoskeletal proteins that we've talked about before in the class, actin and myosin, and those arrays of cytoskeletal proteins and their interactions with one another are what allow this muscle cell to contract when stimulated and lead to its ability to carry out that specialized function. Here we see a close-up of skin. So we see layer upon layer upon layer of skin cells forming a nice barrier between our insides and the outside world. And these skin cells are specialized to express a tough fibrous protein called keratin. Gives us a little bit of a stronger, tougher barrier layer between us and the outside world. But clearly, just looking at these different cell types, they look different, they have different physical properties, they have different structures, and that allows them to carry out these different functions that they serve in the body. Well, how is it that they have these different characteristics, these different features in the first place, right? So first of all, we said that they're all produced by mitosis. So They're produced by mitotic division, and as we know, mitotic division results in clones. So they all have the same DNA. And this is true for nearly all cells in the body, with only a couple of exceptions. They all have the same genetic information, so it's not that neurons have a different set of genes or a different set of instructions than muscle cells or skin cells have, but rather it's that these cells are expressing different proteins because they are expressing different sets of genes. This is something that we talked about in the previous topic, this idea that different cell types in the body, or even the same cell type under different conditions, control which genes out of that instruction manual are being expressed at any given time in a number of different ways. So these cells are different from one another because they express different sets of genes, and that leads them to have different proteins and other biomolecules within the cells, and those different proteins lead to different characteristics that these cells have, and therefore different specialized functions, right? Makes sense. Control gene expression, you control the proteins that are expressed, you control the physical and structural properties of that cell and how it interacts with its environment, and therefore you get specialized functional properties of those cells. As we discussed in the last topic, there are a number of different ways that cells can control gene expression, a number of different levels at which that can occur, everything from regulating transcription to regulating the proteins themselves. But it turns out that during development, this specialization of cells into different cell types relies primarily on transcriptional regulation. Most of the processes of differentiation that occur during development occur because different sets of genes are being turned on or activated or turned off, repressed. To get a better understanding of how this transcriptional regulation actually works down at the molecular level, we're going to take a look at two specific cell types that express different sets of genes, the pancreatic cell and red blood cells. So here we have a pancreatic cell, and this particular pancreatic cell is a cell type called the beta cell that's specialized to produce a hormone called insulin, which I'm sure you've heard of. It's a hormone that's involved with regulating glucose uptake into cells and therefore regulating glucose levels in the bloodstream. And here we have a representation of an immature red blood cell, which, as we just discussed, is specialized to contain lots and lots of hemoglobin protein, right, that allows it to transport oxygen throughout the bloodstream. But before I go on, I just want to briefly mention why I say immature red blood cell, because it turns out that red blood cells are one of the exceptions to this rule that all cells in the body have exactly the same DNA. Red blood cells in the process of maturation actually get rid of their entire nucleus as well as their cellular organelles, and they're literally just big bags of hemoglobin floating through the bloodstream. So we would be talking about the gene expression that occurs in an immature red blood cell on its way to becoming a mature blood cell that would be circulating in the bloodstream. Now, if we looked in the nuclei of these two different cell types, we would see the same set of genetic information. So both of these cell types would have an insulin gene and they would have a hemoglobin gene, right, represented here in the blue. This is the actual genetic information. Both of those would have, as we discussed previously, a promoter that serves that gene, right? And they would both have these transcription factor binding sites that we talked about in a previous topic, which is where those proteins called transcription factors that regulate gene expression at the level of transcription bind to specific sequences in the DNA, and by interacting with the RNA polymerase that's bound at the promoter, generally in us is activating gene expression. And so the insulin gene has a particular set of transcription factor binding sites. The hemoglobin gene has a particular set of transcription factor binding sites, and those are responsible for the appropriate expression of those genes. And by appropriate expression of those genes, of course, I mean that regulate hemoglobin in such a way that it's expressed in red blood cells but not in pancreatic cells, and that regulate the insulin gene such that it's expressed in pancreatic cells but not red blood cells, right? So how does that occur? Well, it involves these transcription factor binding sites, and more specifically, the proteins that interact with them, those transcription factors. So in order for that insulin gene to be expressed, the transcription factor proteins that bind at each of those sites all have to be present, right? The entire set of transcription factors that are required to activate that gene have to be present and expressed. So it's not just one transcription factor, it's actually a whole set of transcription factors. I'm just going to abbreviate that TF. The right combination of transcription factors must be present in order to get that gene expressed in that cell type, right? So it's a combination of different transcription factors, and that's referred to as the combinatorial code. It's like having a numerical code to the safe. It doesn't do you any good to have 2 out of the 3 numbers that you need. You need the entire code in order to crack that safe, and you need the entire code in order to unlock gene expression in this case. So the reason that the insulin gene is expressed in pancreatic cells but not in red blood cells is that in pancreatic cells we have exactly the right combination of transcription factors that bind at those sites and can activate insulin gene expression. However, in pancreatic cells, we do not have the right combination of transcription factors to activate the hemoglobin gene, right? We may have 1 or more transcription factors, but we don't have the entire code, we don't have the entire set, and so that gene doesn't get expressed. And of course, in the red blood cell, then the situation would be reversed. We have the correct set of transcription factors present that are required to activate the hemoglobin gene, but we don't have the required set to activate the insulin gene, and so that gene's not expressed in red blood cells. So it's a combination of specific transcription factors that leads to specific gene expression, and that specific gene expression is what leads to those very distinct structural and functional properties associated with these different cell types. But I can hear you asking the question, well, how did these cells come to express these different sets of transcription factors? They all came originally from the same fertilized egg, so at what point in development did they start expressing different proteins? Did they start expressing these different transcription factors so that they could turn on different sets of genes that allowed them to become specialized in this fashion? That process of activating specific sets of transcription factors and causing specialization of cell types actually occurs very early in development. So let's take a look at the earliest steps of human development to get an idea of how that transcriptional switch starts getting flipped. So the process of human development obviously starts with fertilization, the union of ova or egg and spermatozoa or sperm, and that union results in fertilization and the formation of what we now call the zygote. So that zygote is a term that's used to refer to the earliest stages of embryonic development. And for about the first day or so following fertilization, from the outside you wouldn't see much happening in this zygote. On the inside there's a lot happening. So the entire genetic program is being reset from what it was in these haploid germ cells to what it needs to be in this zygote, this early embryo that's going to be undergoing very rapid mitotic division and ultimately a differentiation of these cell types. So it's resetting the genetic program to allow that to happen. But following about 30 hours, what you'd start to see happening would be cell division. And the way that this happens is in this very characteristic way. The outside of this zygote is actually covered with a little membrane. It's not the cell membrane of the ova itself, it's outside of that cell membrane. And as cell division occurs, the newly formed cells are contained within that. You might think of it as kind of a little bit of membranous shell, right? So these divisions produce new cells. But the interesting feature about these new cells that are being produced is notice that they don't grow in size, right? They're contained within this membrane, and the cells actually get smaller and smaller with each cell division, right? So ultimately, you end up with this little ball of cells that's contained within this membrane. And this earliest set of cell divisions, this earliest process of development, is referred to as cleavage. It's cell division without cell growth. And that leads to an increase in cell number, but a decrease in cell size. Which actually makes sense because the human ova is actually a pretty large cell. You can actually see it with the naked eye. It's about the size of a period at the end of a sentence. So it's unusual in its size, and we know that the average cell in our body is about 10,000 times smaller than that, or about 10 micrometers in diameter. The end product of cleavage, this little ball of cells, is called a blastocyst. And this whole process takes about 5 days following fertilization, start to finish. If we zoom in and we take a look at that blastocyst in a little bit more detail, what we would see is something that looks like this. So if we looked at it in cross-section, we would see that there's kind of an external layer of cells all around the outer surface of that blastocyst. So again, we're looking at a cutaway view at what this blastocyst looks like on the interior. And then we would see a cluster of cells all tightly compacted together. And this stage actually marks the first time during development where the cells in this early embryo actually have different fates, right? So these cells that make up the outer layer of the blastocyst are going to form what are called extra-embryonic tissues. And by extra-embryonic, I mean outside the embryo. So these are tissues that are important for supporting the pregnancy, providing nutrition and gas exchange for the growing embryo and fetus. Things like the placenta, right? So that would be extra-embryonic tissues. Whereas this little ball of cells in the interior, what's referred to as the inner cell mass, these cells are actually going to form the embryo itself. So by this very earliest stage of development, which is again just 5 days post-fertilization, these cells, the cells that make up the outer layer, and these inner cell mass cells have different fates already, right? So they're already fated to become different things. But an important thing to note here is that all of these cells in the inner cell mass have not assumed different fates at all. Every one of these cells is capable of forming every single one of the tissues that are ultimately going to make up that embryo. These are referred to as embryonic stem cells. And we're going to talk about stem cells a little bit more in detail in the next topic, but just kind of set that up as to where that term comes from, embryonic stem cells. These are embryonic stem cells. They are the inner cell mass cells of a blastocyst that exist for a few days very, very early in development. So these outer cells have a different fate from these inner cells, but all of these inner cells are fated to become parts of the embryo. They just haven't specialized in any way for different cell or tissue types. What happens next in the process of development is that these external cells start secreting fluid. So this whole cavity starts to fill with fluid, and we start to see more proliferation of these inner cell mass cells, so more cell division occurring. And ultimately what we are going to end up with at the next stage is something that looks like this. And I'm not going to draw in these cells that are on the outer layer. I'm just going to draw in these cells that make up the inner cell mass. Because of the fluid that gets produced by these outer layer cells, that inner cell mass gets kind of pushed to the middle. So it forms kind of a little flattened disc that's made up of multiple layers of cells, kind of piled up on top of one another, stretched across the middle of this. And of course there'd be that outer layer of cells that we had over here. I'm just not drawing them in. Now what happens next is that we're actually going to see this primitive embryo undergo the next stage of development, which is called gastrulation. Gastrulation marks the first time point in development where we see differentiation of these inner cell mast cells into specific tissue types occur. And these specific tissue types that are generated by the process of gastrulation are the primitive, what are called germ layers that are going to produce all of the tissues of the body. What happens at this stage of gastrulation that causes cells to start to differentiate into different tissue types is that gene expression within these cells starts to change. These cells occupy different positions within this tissue layer. They have different interactions with the outside world, right? The cells that are at the interior here are bounded on both sides by other embryonic cells, whereas the ones at the surface layer are in contact with the fluid-filled cavity that they're right next to. So they have different immediate external environments. They have contact with different types of cells and potentially chemical signaling molecules. And we start to see changes in gene expression even at this very early stage. If you look at different cell types within this embryonic disc at this stage of gastrulation, which, you know, again, we're talking about maybe 12 to 14 days in after fertilization. So this is very, very early in the process of development. We would already start to see changes in gene expression causing differentiation of cells into specific cell types. Now, they're not very differentiated at this stage. As I said, what they're going to be forming are kind of primitive precursor tissues that are referred to as the germ layers. So if we zoom in and we look at this layer of cells that's here in this little embryonic disc in a little bit more detail, we would see something that would look kind of like this in that flattened disc. So you'd see 3 layers of cells, top layer, a middle layer, and a bottom layer. So again, there'd be cells in here. I'm just going to color code these cells to show that they are in fact differentiating into different types of cells already at this early stage. So these middle guys will color green. And then this bottom layer will color yellow. So these are cells, but they're already different from one another in terms of what type of tissue they are. So this top layer that I've kind of labeled in blue here is a germ layer that's referred to as ectoderm. And that layer of tissue is going to go on and ultimately form the nervous system tissues and outer layer of skin. This middle layer is referred to as the mesoderm, germ layer. And the tissue types that result from this mesoderm layer are muscle, bone, blood, and then finally this bottom germ layer is referred to as endoderm. And endoderm is responsible for producing gut as well as most of the tissues that are found in lung. So the changes in gene expression that have occurred at gastrulation have resulted in differentiation or specialization of the cells within these 3 layers so that they already have slightly different properties, and they're definitely going to have different fates within the embryo as development proceeds. What happens during this process of gastrulation then is that these cells start to express different transcription factors. And those transcription factors turn on specific genes and these specific genes ultimately lead to the development of specific cell types that are going to be part of tissues and ultimately organs and organ systems, as we know, making up the fully formed human organism. And this final process of development takes a lot more time. We're not going to spend much time talking about the individual steps of it. Is referred to as organogenesis, right? The origination of the organs and organ systems. But all of these steps, all of these specializations rely on transcription factors that activate specific gene programs that result in distinct cell types being formed and therefore different tissues and different organs being present within the body. To give you a better overview of what happens during these earliest stages of human development, I'm going to show you a clip from a YouTube video. This is a presentation that was done by Dr. Douglas A. Melton as part of the Howard Hughes Medical Institute's 2006 Holiday Lectures on Science series. His talk is a very nice in-depth presentation regarding embryonic stem cells. This clip is just one small part of it, but I would encourage you to go to YouTube and watch the rest of it if you're interested in the topic. So what we're seeing here, we're going to be zooming in on the ovary. We're going to see the process of ovulation. Here's an egg being released, traveling down the fallopian tube, and here are millions of sperm traveling up the fallopian tube. One of those sperm is going to unite with the egg in the process of fertilization. And as I said, during this earliest period, the first 30 hours or so, we don't see a lot from the outside, but inside there's reprogramming of that genetic information going on. Following about 30 hours, we start to see cleavage occur, and that's those rounds of cell division in which we get more cells, but the cells actually get smaller and smaller with each division, eventually yielding a little ball of cells, anywhere between about 50 and 150 cells by the end of the cleavage process. And that ball of cells we refer to as the blastocyst. And if we take a look at the interior of that blastocyst, we see that inner cell mass that's going to be destined to become the embryo proper, that outer layer of cells again becoming extra embryonic tissues like the placenta. We see implantation into the wall of the uterus and further development. So here's that next stage where we see proliferation of those inner cell mass cells. And they migrate to the middle, that fluid-filled cavity, and form this disc. And the process of gastrulation begins, where we start to see proliferation of cells, movement of cells. They start to migrate within that disc. And we see formation of those three germ layers, the ectoderm, mesoderm, and endoderm, that I showed you in the drawing. Then those germ layers start to fold up in characteristic patterns. And eventually we start to see the formation of primitive organs and limbs, and then ultimately what looks more like the embryo that we're used to seeing. There's a little bit more detailed view of those different germ layers and the specific adult tissues that they're associated with, ectoderm with nervous system and skin, that inner mesoderm layer associated with muscle, bone, blood, kidneys, cardiac muscle tissue, and then finally endoderm associated with the gastrointestinal tract. That's everything associated with gut as well as most of the tissues of the lungs. We can see even at this very earliest stage of embryonic development some of the precursors that organogenesis is going to transform into functioning organs. So this little yellow highlighted lump of tissue here to the side of this endoderm is the pancreatic bud. It's eventually going to form the pancreas. This little bud over here is going to become the liver. This will become stomach, and this little bud here will become lungs. And that process of transformation in which these primordial tissues are converted into fully functioning organs is that process of organogenesis that I referred to earlier. But how does this little lump of tissue that's eventually going to become the fully formed functioning pancreas get transformed into that complex organ? Well, it's driven by a set of gene expression patterns that are put in place by specific sets of transcription factors that drives a particular set of growth patterns here, cell proliferation events and tissue folding events. It ultimately leads to that organ taking on its final shape with its specific population of cells carrying out their specific functions. And so that one function that we talked about earlier, the expression of pancreas-specific gene insulin, would be just one example of a tissue-specific gene program that's set in place during the process of organogenesis. One thing that's been recognized for a really long time, it's actually something that Darwin wrote about in his book on the origin of species, is that if you look at different species of animals at early stages of embryonic development, it's very difficult to tell them apart. They're really similar at early developmental stages. You can see a nice illustration of that right here. We've got 4 different embryos. These embryos are human, cat, cow, and horse, but just to look at them, it's really hard to tell them apart. They're structurally very similar at this stage, and that's because what's going on at this stage is very similar no matter what species you're talking about. You're talking about specifying, a head versus tail regions. You're talking about specifying where the limbs are going to be, setting up a basic body plan, an axis of symmetry, right from left, top from bottom, front from back. And those developmental steps, those simple developmental steps that occur early on, happen very similarly no matter what species that you're looking at. So that's been known for a really long time, but it's only with recent technology where we were able to actually get at the gene sequences that are responsible for these earliest stages of development that it became apparent that the reason that these developmental stages are so similar across species is because the genes that regulate these early stages are extremely highly conserved across species, meaning that they can be found in all these different species, either the same gene or very highly related genes. And it's the developmental steps that are initiated by those commonly expressed genes that cause these stages to be so similar no matter what species you're looking at. So if you're interested in knowing which of these embryos is which, maybe you guessed, human is D, cat is A, the cow is B, and the horse is C. And actually, if you take a closer look at that end of that forelimb on that embryo B, you can see that it looks more like a hoof than a hand. So there are some differences that can start to become clear as these embryos get further and further along in development. It's only at these earliest stages that they're so similar. An excellent example of this kind of conservation of developmental genes are in what are called the homeotic genes, or what are sometimes referred to as Hox genes. These genes encode developmentally important transcription factors. So these are proteins that regulate the transcription of other genes, and they're referred to as master regulators of development. And the reason that they're referred to as master regulators is because they initiate entire gene expression programs. So they turn on whole sets of genes and initiate gene expression cascades that lead to downstream events that cause the specification of how the body is actually arranged, meaning head to tail, front to back, side to side, as well as the specific development of particular body structures and organs. So these Hox genes were initially identified in fruit flies back in the 1980s, and their role in development was demonstrated in that organism. So this is an illustration of a fruit fly chromosome that contains a cluster of these Hox genes, and again, these genes encode those transcription factors. So these genes would be themselves transcribed, produce proteins, and those proteins would go on to regulate other genes. But the products of these Hox genes are responsible for the specification of the body plan of this organism. And it came as a huge shock to the developmental biology scientific community when it was discovered that if you look in the human genome, you would see clusters of extremely similar genes to what you find in fruit flies, right? With the exception that we don't have just one set of these homeotic genes, these Hox genes. We have multiple sets because our genes have undergone duplication over the course of our evolutionary history. But the really shocking thing was that they function in exactly the same way in humans to organize the basic arrangement of body regions, so top, head to tail, front to back, these sorts of body plan specifications, as they do in fruit flies. And not only are these genes responsible for specifying the arrangement of body regions, but also for specific body structures. And the effect of misexpression or mutation of these genes really illustrates how important they are for determining the overall body plan and how body structures are developed. So in one example of this in fruit flies is a gene called antennapedia. And antennapedia is a Hox gene. It's normally expressed in the thorax of the fly. So I'll just abbreviate antennapedia, ANT. The expression of antennapedia within this specific region of the fruit fly is responsible for and actually drives the gene expression program that leads to legs being formed on that particular segment of the fruit fly. If you misexpress using genetic tricks, instead of expressing antennapedia here in the thorax, if you instead use genetic tricks to cause antennapedia to be misexpressed in the head, then what happens is you see a transformation where antenna become legs. So this gene specifies legs, and wherever it's expressed, it's gonna cause legs to be formed. And the same sort of dramatic effects on body plan or the formation of body structures can be observed with mutations as well. If you knock out the function of these genes, then you see similarly dramatic effects on the specification of the body plan and particular body structures within that organism. You also see that in humans as well. So mutations in Hox genes in humans are associated with birth defects, things like cleft palate, and has been associated with some mental retardation syndromes, developmental delay. Although mutations in humans don't have as dramatic an effect as what you see in fruit flies, and part of the reason for that is that we don't have just one copy of these genes, we have multiple copies, and so essentially we have kind of backup copies of these genes. Their functions are a little bit redundant and overlap a little bit, so we don't see, legs coming out of heads and things like that in humans. You also can see similar sorts of problems, birth defects, and developmental abnormalities with chemical exposure that disrupts Hox gene expression during development. So you probably are aware that there are some drugs or chemicals that pregnant women are supposed to stay away from, and if they don't, then their fetus is at risk for having birth defects. And some of these, the way that they cause those birth defects is by disrupting Hox gene expression. So normal expression of the Hox genes during development is required for the normal developmental progression to occur. So if you have, for example, mutations in the Hox genes that control limb development, so if we go back to our early embryonic stage, at this stage of development we have formation of these what are called limb buds, the early precursors to arms and legs, and the formation of those body structures, like so many, is controlled by these Hox genes, these homeotic genes that initiate gene expression programs that lead to the extension of that limb, the lengthening of that limb, and ultimately to the formation of individual digits within that limb, resulting in fingers and toes. So what happens if you disrupt the expression of Hox genes in that region of the embryo? Well, you'd obviously disrupt. So if you had a mutation in the Hox genes that control the development of that limb bud and therefore the extension of that limb, the formation of fingers, you have a disruption of that in humans, then you end up with a birth defect and abnormal development of that limb. So that would be a very bad outcome for humans, but that very same event has actually occurred and resulted in the formation of new species. So mutation in Hox genes can be a driving force for new species, new variations that natural selection can act on, and therefore an important driving force for evolution. A good example of that is a mutation that did exactly this, but in reptiles, right? So here's a lizard, has perfectly normal limb bud and limb development. It's got a short body, 2 forelimbs, 2 hind limbs. Mutation in that set of Hox genes that controls limb bud development led to snakes. Snakes have a mutation in the Hox genes that control limb bud development, so forelimbs and hind limbs don't develop in snakes as they do in lizards, and that led to, entire new species being formed. So nearly all snakes are lacking forelimbs and hind limbs, the exception to that being boas and pythons, which in these 2 species of snake, the remnants or vestiges of hind limbs are still present. So you see these little what are called spurs sometimes at the tail end of the snake, so it would be right about here. And these actually have acquired a new function. They're used for attachment during mating. But the interesting thing is that if you look at the skeletal structure of the snake, so if you take a look at the bone, what you would actually see is a vestigial pelvis in here, a pelvic girdle for the hind limbs to have formed. It's just that they don't fully develop because of that disruption in Hox gene expression. And we also see kind of intermediate species that seem to be somewhere in between, right? So it's not quite a lizard, not quite a snake. This is a skink, and it's got this kind of elongated body that's characteristic of that evolutionary transition. And the forelimbs and hind limbs are clearly much smaller than what we see in lizards. But, again, that's due to a difference in expression of those genes that control the specification of these body structures. So again, this just highlights the fact that mutations are neither good nor bad. Mutations are neutral, and what determines if a given mutation is favorable is the environment. So for a human that has a mutation that disrupts limb formation, that would be a bad mutation. It would be an unfavorable mutation. But if you're, for example, a burrowing lizard, then maybe having smaller limbs is a selective advantage for you. It helps you get through your burrow quicker and move around easier. If you're, an aquatic reptile, maybe it serves the same purpose. And in fact, you see the same sort of vestigial hind limbs within the skeleton of whales as well.
Well, that concludes our whirlwind tour of developmental biology and the role of transcriptional regulation in controlling the processes that occur that lead to specification of different cell types in the embryo. Now that you've watched this video, you should be able to answer these concept check questions.